Okay, welcome everyone. Welcome to the OTS Trainee Spotlight series. So my name is Ronald Buijsen. Together with Hassan Faki, we will be the host of today. And I think the speakers will introduce themselves, and we will start with Tom Metz, Ph.D. student in the lab of Willeke van Roodlom at LUMC. So Tom, the floor is yours.
Yeah, thank you, Ronald. So as Ronald said, my name is Tom Metz. I'm a PhD student in the group of Willeke van Roodlom at the Leiden University Medical Center in the Netherlands. And today I would like to share with you some of the work we did on looking into the biodistribution of a radioactively labeled AON after ICV and IT injection in mice. So to first introduce the disease that we're working on. So we are developing an AON against a disease called Dutch type hereditary cerebral amyloid angiopathy, DCAA, also known as Katwijk's disease. And this disease originates from a small town called Katwijk in the Netherlands, close to Leiden. People suffer from hemorrhagic strokes, and where the first stroke is fatal in 2/3 of the patients, and recurring strokes happen, and also vascular dementia, and eventually leading to an early death. As mentioned, it is quite rare. Only 400 to 500 people are at risk, estimated worldwide. We're developing an exon skipping strategy for this, because the CAA that happens, the hypothesis is that the accumulation of the protein amyloid beta around blood vessels causes blood vessels to rupture and to lead to this disease. And we are developing an exon skipping strategy on the precursor protein of this amyloid beta protein called APP. There is, in Katwijk's disease, there's a mutation, a C2G point mutation on exon 17. And our strategy is to skip exon 17 from APP, thereby removing the Dutch mutation, and thereby also removing a part of the amyloid beta coding region. And the hypothesis is that this will lead to a decreased amyloid beta formation, and also decreased amyloid beta aggregation, and therefore decreased disease pathology. So for my colleague Elena Dautzali has done quite a lot of work with this AON in cells, so in lots of different cells, but also in human patient-derived induced pluripotent stem cells, and she saw quite a good dose-dependent skip efficiency. So in the top, the full-length APP, and on the bottom, the exon 17 skipped isoform, APP delta 17, on RNA level and on protein level, and it also causes a decrease of amyloid beta in the cells, which you can see in the bottom graph. And then next step was to test this in an animal, and this was in a wild type mouse model. And there it also gave a quite high skip, so about 70% in different brain regions, and also on protein level a 40% skip or C. So then, because we inject in mice, we inject these AWANs directly into the right lateral ventricle, and because of directly injecting into the CNS, you surpass the blood-brain barrier, and this has some major advantages compared to more peripheral targets for AWANs. And we have previously in our lab, Lodewijk Tonen published this, showed that up to 3 months the AWAN will stay in the brain. But because clinical applications of AWANs, they will not be ICV injected, because this is quite invasive. So these clinical applications so far have been mostly done intrathecally, so IT. And with this research, so we want to see how does our AWAN distribute throughout the body of a mouse in the first hours and days after the injection, and how does the injection method affect the distribution of the AWAN. So for this we used SPECT imaging, so single photon emission computed tomography. So an AWAN is labeled and with a radioactive label, and then gamma ray photons are emitted by this source, and they are detected by a set of collimators in the SPECT machine that you see here. And the advantage of this is that you can do longitudinal measurements in the same animal. And you can measure not only the brain, but you can measure the whole body of the mouse. So for this we have, this is a graphical depiction of our AWAN, and this is the AWAN with DOTA attached, and this DOTA acts as a sort of pocket for the radioactive label, which is covalently bound to the AWAN. To check if this affects the exon skipping efficiency, we first tested this on cells. So on the left is the AWAN, and on the right is the AWAN with the DOTA attached. And here on the top is the full length, and on the bottom is the APP delta 17 isoform, and the DOTA attachment does not seem to interfere with the exon skipping effect. So then we continue to label the AWAN with radioactive indium 111, which has a half-life of 2.8 days. So we made this on the day of injection, and then injected mice either intrathecally or intracerebroventrically. So on day 0, and then immediately after this, we did the first SPECT scan, and then a second scan at 4 hours, and then at 24, 48, 72, and 96 hours. And these mice were about 3.5 months of age, and both male and female. And for the so for ICV injections, we do this standard in our lab with the cannula guide, which is placed at a specific location which targets the right lateral ventricle. and then AON is infused through this infusion cannula. And if you do multiple injections, you can attach it to the skull, but in this case we do not attach it to the skull, and we just suture the skin. On the right, you see an MRI image where you can see the cannula entering the right lower ventricle. For IT injections, I had no experience with this, so I, together with Ernst Zuidgeest, we looked into some older literature how to do this, and so we found quite a good method that worked quite well. We tried this with ink, and yeah, so we saw that it was the ink was throughout the spinal cord and also into the brain, into the ventricles. So then we proceeded to inject the animals with AON. The average dose was 220 microgram, and the average activity of each injection was about 31 megabecquerel. And in this table, you see the injected activity that is measured in the mouse directly after injection. As you can see, it differs quite a bit between the ICV and IT injection. The ICV injection gave a much higher activity, and for the lowest two, they were quite much lower than the rest. So we excluded those from further analysis. So then the mice were measured for activity before each scan, before each SPECT scan, just total body activity, and this already shows that that the signal decreases over time, and after 96 hours, about 75% of the signal is excreted from the body. So, and then if you look into the SPECT scans, so these are the scans from the ICV injection. So on the left is the first scan, and then you see that it nicely distributes throughout the whole brain. So this is, I should say, the sagittal section of the mouse. So here you see the brain and the beginning of the spinal cord, and in after 4 hours you see it spreading throughout the whole CNS, and also in the kidneys. And what you can see if you look at it over time is that the signal in the brain stays quite stable, while the signal in the kidney goes down quite rapidly, indicating that that when the AON is in the brain, it's quite stable. Yeah, so another thing you might see is that this dot here is the thyroid, and this is where unbound label accumulates. So this is due to breakdown of the label from the AON. Then for the IT injections, you see, so this is the injection site. You see it's spreading quite locally in the first scan, and then after 4 hours, you see it also in the kidney. You see it spreading throughout the CNS, just like with ICV, but not as much into the brain. And then yeah, you see the same thing. It stays in the CNS, but in the kidneys and in the rest of the organs, it goes down over time. So then afterwards, we collected all different organs to measure their radioactivity. And So what you can see here is in dark blue the ICV injected, and in light blue the IT injected. Overall, the pattern is quite similar. You see a lot of signal in the kidneys and liver and gallbladder. The other organs are quite low, and the biggest difference is in the brain. So there's with ICV injection, there's a lot more in the brain. So if this is the same graph, but then in a log scale, and this is just to show that indeed it gives quite a similar pattern also in the more lower in the organs with the lower signal, and you do see a clear effect in the brain that there's with ICV injection more in the brain. And if you zoom in to the brain, into the different brain regions, you also see this difference, except for the spinal cord. And this again is in log scale. And then, yeah, you see that IT injection has lower signal in the brain, but also overall the same pattern. So there's more in the olfactory bulb and a bit less in the cortex. So yeah, to conclude from this, so how does the distribution of our AON go in the body in the first 1,000 days after injection? So we see the signal in the kidney going down, while in the brain it's quite stable, indicating that that it's the AON is excreted over time, but stays within the CNS for longer than 4 days at least. And we do see that the injected dose in ICV is higher than in IT injected. And this also leads to a higher signal in the brain. But the overall distribution pattern seems quite similar. And also this distribution pattern is also comparable to some other, to another paper that did the IT injection of a Gapmer AON in a rat. This gave quite a similar spread. They did not compare to ICV. Yeah, so one thing is that these IT injections in mice can be quite difficult, and also because the size of the spinal cord is not as big as in humans, and there's a limited volume of CSF where you inject into compared to the lateral ventricles. And this could be an explanation for the lower signal in the whole mouse and also in the brain. Because the overall distribution pattern does seem similar between IT and ICV. And so what we're doing now is to analyze the exon skip levels on RNA level in the brain as a confirmation. And we are trying to set up a splint ligation assay, which is a new method. Was published by John Watt's lab. We want to use this to try to confirm this AON distribution. Yeah, so that was it. And I would like to thank my research group, so Willeke van Roon's research group, especially Willeke and Louise van der Weert for their supervision. And yeah, Mick Welling is the one who helped me with the SPECT scanning, and Ernst Zuidgeest with the injections. And Esme Nieuwenhuizen is a student in our lab, and she's working on the RNA analysis. Yeah, so that was it. Thanks.
Thank you so much, Tom, for this presentation. Very interesting work. So we have a couple of questions for you. So I will start with a question from Jeffrey. who was asking about the chemistry of the ASO that you've used for the exon skipping, and maybe you can also comment if you have tested different chemistries as well.
So it's a 22-mer, and it's splice switching AON with to MOE modifications and a phosphatidylate backbone. And we did not test any other chemistries. No.
Okay, so we have another question from Bruno, and I'm going to combine like both of his questions in one. So basically, he's asking, how much is the radiolabeling affecting the distribution of the oligo, do you think? And did you, for example, try to inject a radiolabel control to understand if like it has a similar kinetics to an oligo attached to them?
I didn't quite get the first part, sorry.
So he was asking, how much do you think that the radiolabeling is affecting the distribution of the...
So I would say that it doesn't, I think it would really be a minor effect. Yeah, so as I showed before, we did test it on cells to see if the DOTA in the linker attached to the AON have an effect. They don't seem to have an effect on the exon skipping in cells. Of course, you don't know how it affects the distribution in a mouse, but it's really small compared to the size of the AON. If you compare it, for instance, with studies that use immuno labels, they are much bigger. So I think it would be a minor effect.
Okay.
And we are now doing this splint ligation with unbound AON to compare.
Okay. Yeah, exactly, because it's good that you did the activity comparison, but I believe that distribution could be another story.
Yeah.
So we have another question from Aurelie. She says, thank you for the great talk. And she was asking if you directly measured the quantity of the oligo. And I believe that's what you're working on now with the splint method. She's asking if you try to measure directly the oligo rather than just the radioactive signal.
No, we did not do that, but that's what we try to do with the splint ligation assay.
Yeah.
And then and we try to measure the functional effect by measuring the exon skipping in the brain regions. Yeah.
Okay, and we have another question from Min Sung Park, who's asking how much volume or concentration of the injected oligo you did for both ICV and IT?
Yeah, so for both injected 10 microliters, and with a total of on average it was 220 microgram. It was slight variations, but it was about 220 microgram.
Okay, great. I will take one more question, I believe. So a follow-up by Bruno is that he noted that the linkage between the AON and the label, PS linkage, he's curious if you think this is being cleaved over time?
I think it will eventually be cleaved, but yeah, I don't think it will be cleaved like that fast. But yeah.
Okay, perfect. Thank you so much, Tom. This was very interesting. Yeah. There's more questions in the chat if you would like to take a look.
Okay, yeah, I'll have a look. Okay, thanks.
And for the sake of time, we're going to be moving to our second presentation of today, by Briana Wilson, who is going to be talking to us about how XRRN2 prevents the entry of tRNA trailers into RISC. So, Briana, the floor is yours.
Thank you very much. And yeah, I agree. Excellent talk, Tom. So my name is Bree, and I am at the University of Virginia in the Medical Scientist Training Program. I'm nearing the end of my graduate school years in that program. I'm actually in the Dutta Lab, which has relatively recently moved to the University of Alabama, Birmingham. And thank you for allowing me to speak today. So as you can tell by my title, I'm going to be discussing tRNA fragments, specifically tRNA trailers. And this is a relatively new area of research in terms of small RNAs. And this has really come about from an unbiased view of where small RNA reads map from small RNA sequencing. So, for example, if you look at small RNAs from K562 cells, about half of them map to canonical microRNAs. But the second largest class of small RNAs are actually mapping to tRNAs. And these small RNAs are about 18 to 30 nucleotides. So it turns out that we still don't have a very good understanding of how these small RNAs are being created. I know this is a very busy slide, but this is just to show that there's some evidence that supports dicer as a biogenesis enzyme producing some of these tRNA fragments. However, you can knock out dicer and still get an abundant amount of tRNA fragments. For the tRNA fragments that I'm going to discuss today, the biogenesis enzyme is actually known. So, for example, going from a precursor tRNA to a mature tRNA involves cleavage of a leader strand by RNase P. A trailer strand has to be removed, and that's done by RNase Z or ELAC1 or 2. And precursor tRNAs may also have introns, so those need to be removed as well. So this is this cleavage of the trailer produces TRF1, so you'll hear me refer to those tRNA trailers as TRF1s in the rest of the talk. So a lot of different functions have been attributed to tRNA fragments from post-transcriptional gene silencing in the case of TRF3s, to translational silencing for longer tRNA halves. TRF1s have not been attributed a sort of microRNA-like function, which I'll get into a little bit later, but has been shown to bind certain RNA binding proteins, and there is an ability for precursor tRNA to be funneled into the microRNA process if certain maturation factors are not present. And initially, my lab started characterizing TRF3s and really focused on their function in the microRNA-like pathway. So speaking of microRNAs and RNAi, it took 20 years from the initial RNAi discovery for us to develop RNAi therapeutics and get them to patients. And I think this is a really fascinating process, and I see a lot of potential for tRNA-derived small RNAs to also be used in the clinic. So it actually turns out for the TRF timeline, we sort of became aware that these TRFs may be present back in the 70s. So it was noted that modified nucleosides were present in the urine of cancer patients. It wasn't until 2009, 2010 that there was more rigorous characterization of tRNA fragments. And then in 2018, my lab actually elucidated this microRNA-like function for TRF3s Another lab, Christian Belotti's lab, found that specific modifications of TRFs is important for translation in hematopoietic stem cells. And just last year, it looks like a startup has been founded based on those findings from the Belotti lab. This doesn't seem like it's an actual TRF therapeutic, but instead they're working on developing inhibitors of the modification enzyme. So we're not quite there yet with TRF therapeutics. And part of the reason I think that is that we're still missing some fundamental understanding of TRF and tRNA half biology. So as I mentioned before, we don't really have a good understanding of how these tRNA fragments are being produced. And I wanted to highlight that because tRNA therapeutics is a very hot area of research, and there are startups trying to develop these, but I would maybe warn that if we don't know how tRNA fragments are produced, these tRNA therapeutics may produce some tRNA fragments that may have some off-target effects. And then we don't know anything really about the stability of endogenous tRNA-derived fragments. And I think that's one of the major triumphs of RNAi therapeutics is our understanding of microRNA stability, and also how we can improve siRNA therapeutic stability. So the focus of my talk today is, first of all, on a sort of global unbiased view of small RNA stability, specifically TRF stability, and then how there might be certain features or certain proteins that may be regulating TRF stability. So for example, I'll talk a little bit about an exoribonuclease called XRN2 and show evidence that supports its role in keeping TRF1s highly unstable and out of actually the microRNA pathway. I think this has potential to help us sort of rescue certain types of small RNAs that we think might not be functional, but if we can understand how they're regulated in the cell, then we can give them novel functions and perhaps improve their therapies. So there's lots of hidden treasure in publicly available data. And I actually didn't have to do any small RNA sequencing myself to start understanding TRF stability. Folks have been interested in microRNA stability for quite some time now. And this study from the Lu lab looked at microRNA stability in an AML cell line, HD-MYZ. But as I said before, TRFs are about the same size as microRNAs. So I can basically find any small RNA that's about the same size as microRNA and get a good idea of what its stability is. So after taking an unbiased view of where these small RNAs are mapping, what I found was, first of all, microRNAs were quite stable. So the time course of actinomycin D treatment went up to 12 hours, and most microRNAs were stable for up to 12 hours, or at least 12 hours, which is known. MicroRNAs are highly stable. Some of these other small RNA classes, so small RNAs that map to long non-coding RNAs, there's maybe a wider distribution of stability here. rRNA fragments are generally quite stable, actually, which I won't discuss here, but is quite interesting. And then TRFs, as you can see, also have a wide distribution of stability. There are some that are really quite stable, and then others that aren't so stable. So what's going on here? So TRFs can be broken up into different subclasses based on what part of the tRNA they come from. So these leader sequences, which there aren't very many of because they're actually pretty short and they're not captured in standard small RNA sequencing pipelines. There's one that's highly unstable. TRF5s and 3s, for example, are moderately stable. There are a few species that are quite stable. TRF3s without a CCA, so a CCA is required during maturation, and that's required for amino acylation of tRNAs. So actually, TRF3s that are coming from the 3' end that don't have a full CCA tend to be more stable, it seems. And then honestly, what sort of stood out to me was the fact that TRF1, so those tRNA trailers from precursors and internal TRFs are highly unstable. And you might have looked at that plot and said, well, you only have 6 TRF1s, what's going on there? Every tRNA produces a TRF1. It just turns out that most of them didn't make it to the end of the time course, they were degraded. So to get a better look at what's going on with the majority of TRF1s, I looked at the 0 hour and 1 hour time point of actinomycin D treatment in the cell line. And as you can see, on the y-axis, I have the steady state level of these small RNAs. And as you can see, there's a huge decrease in TRF1s by only the 1 hour time point, while the other small RNA types are quite stable at this time point. And again, here's just a couple of examples of some TRFs from this cell line. And you can see that TRF5s are, this example of a TRF5 is quite stable. This example of a TRF3 is quite stable, but this TRF1 really drops off and drops off very rapidly at the first hour. We were able to validate TRF1 instability in our own cell lines in the lab. So I looked at 293Ts and did qPCR for TRF1001, which is a very highly expressed TRF1 in the cell line. And you can see within 1 hour, it's almost completely gone. The next question I wanted to ask was, can we identify the enzyme or enzymes that are responsible for this turnover? And I wanted to do this because I remember this piece of data from our lab back in 2014, where we looked at where TRFs go in terms of entering argonaute complexes. So basically, this is a per clip reanalysis, and we could see microRNAs entering all 4 different argonaute proteins. And we could surprisingly see TRF5s and TRF3s in AGO1, 3, and 4. However, when we looked for TRF1s in these argonaute complexes, they were completely absent. So I wanted to see if we could find the enzyme responsible for TRF1 degradation. Maybe we could then see TRF1s entering AGO and perhaps behaving like microRNAs. So in order to identify the enzymes responsible for turning over TRF1s, I first looked at enzymes that had been implicated in microRNA turnover. And luckily, just after a couple of knockdowns, I was able to see by knocking down XRN1 and 2. So we did these knockdowns as sort of family knockdowns to first make sure there was no compensation by other family members. And by knocking down XRN1 and 2, I could see a nice increase in this TRF1. And the increase in the TRF1 does appear to be an increase in stability, because when I knock down XRN1 and 2 and treat these cells with actinomycin D for 1 hour, I do see enhanced stability at the 1 hour time point. So that was just TRF1001, but is this a global effect? So what we did next was small RNA sequencing after knocking down XRN1 and 2, and we could see a pretty specific increase in TRF1s. And we could also see a specific increase in these NTRFs, so If you remember earlier, I said these were also really unstable. While, generally microRNAs, they go up and they go down, TRF5s and TRF3s, some may or may not be affected by XRN1 and 2 knockdown. But the real effect seems to specifically affect these highly unstable classes. Excuse me. So far, I have been knocking down XRN1 and 2 together, but is one enzyme more important than the other? So to answer that question, I knocked down XRN1 and XRN2 separately. And what I could see was, it seems like just knocking down XRN2 is sufficient to increase the TRF1s in the cell. And I also wanted to test if the catalytic domain of XRN2 was important for TRF1 stability. And to do that, I looked at TRF1 levels in these cells that express constitutive knockdown or constitutive shRNA against XRN2. And then they have a DOX-inducible wild-type XRN2 or a catalytic dead version of XRN2. So first of all, if you just compare the 293 T cells, you can see an increase in TRF1001 when you knock down XRN2, which is consistent with what we've seen before. And then if you then turn on this wild type XRN2, you can see a decrease in that TRF1001. And then if you express the catalytic dead version of XRN2, you get a rescue of that TRF1001 expression. And none of this appears to be an effect on the precursor tRNA. So from this, it seems like XRN2, specifically XRN2's catalytic domain, is the important part for TRF1 degradation. So now that we've figured out how to stabilize these TRF1s, we wanted to know if that was enough to have them enter Argonaut. And it turns out that it is. So we performed RNA immunoprecipitation and sequencing. So we tagged AGO2, pulled it down after knocking down XRN2 and Cy-selected and performed sequencing. And as you can see, TRF1s, so on the x-axis here, I have the log 2 fold change within AGO after knocking down XRN2 compared to control. And you can see that there's a general increase of TRF1s in Argonaut complexes that is specific. So there's no big changes in microRNAs in AGO2, TRF3s, or TRF5s. And here's just an example of one TRF, 1015. As you can see, an input knocking down XRN2 increases this TRF1. And we also see a nice increase of this TRF1 within AGO after knocking down XRN2. And this is far beyond what's going on in our control IP, our MIC IP. And we were able to validate these findings by Northern blotting. So finally, we wanted to ask if these AGO2 resident TRF1s are actually functional. And to test that question, we knocked down XRN2 and performed mRNA sequencing this time. And we predicted TRF1015 targets and asked whether or not they were more repressed compared to non-targets. And so this CDF plot is just showing that indeed, in red, the targets have, in general, a more negative log 2 fold change compared to non-targets. And then we were also able to see repression of 1015 reporters, luciferase reporters that had complete base pairing with endogenous 1015. So in conclusion, it seems like, identifying the enzymes responsible for small RNA turnover, and then targeting them might be able to enhance their RNA-like functions. But specifically, so it seems like the small RNA stability sort of varies by class, and what's controlling that, we don't know per se. There may be certain modifications. There may be the fact that TRF1s are generally single-stranded. And then TRF1s specifically are highly unstable and degraded by XRN2. And if we deplete XRN2, we can then stabilize TRF1s, and this allows them to enter AGO and repress gene expression. So as I mentioned before, modifications might be playing a role here for other classes of small RNAs like TRF3s or TRF5s. So we're currently working on looking at correlations between modifications and stability. We are also interested in identifying other functions of stabilized TRF1s. So folks have implicated TRF1s in controlling the translation of IRES containing viruses, which I think would also be interesting from a therapeutic standpoint. And we're also testing putative XRN2 inhibitors. So thank you all for listening, and thanks to my lab, specifically the TRF team. So Dr. Zhangli Su and Dr. Pankaj Kumar. I'd like to thank the David Bentley lab for the XRN2 mutant cell lines and my funding. And also just a plug for the Dutta lab. We are looking for some postdoc fellows to work on TRFs, and I'm happy to take any questions.
Thank you. Yeah, this works better. Thank you, Brie, for this excellent presentation. So I think we completely switched gears from something very translational research that might be quickly applicable to a clinical application to something very fundamental. But I think this is very important work, because if you do not do these kind of experiments, you never end up with a clinical application. So I think my very first question is about this. So what do you think is needed to bring this to the clinic? Because probably there are many steps, but what would be the most important step?
Yeah, that's a really good question. I think that understanding what's driving the stability of these guys is important. Not so much for TRF1s, but things like TRF3s, which have been shown to function like microRNAs. It's unclear if all TRF3s are behaving just like microRNAs, or do they have slightly different mechanisms. Some of these TRF5s that can inhibit translation, I think there needs to be a better understanding of the exact mechanism of translational inhibition. And then I think the big thing here is understanding how these TRFs are produced. So yeah, still a lot of basic fundamental things that needs to be elucidated.
Yeah, but very cool experiments. So we have a few questions in the chat. So I will start with a question from Bruno. He says, nice talk, Brie. Would love to hear your thoughts on what you think the advantage of using TRF versus an siRNA for therapeutic gene silencing would be.
Yeah, so thank you, and great question. So folks have been interested in single-stranded siRNAs for quite some time. And the real bottleneck there was that they were highly unstable. You had to use a ton of them to get them to work. So I think the benefit there is that you don't have to worry about the other strand, right? You don't have to worry about if the other strand is going to enter AGO and then regulate genes or what have you. So yeah, I think a single-stranded approach might still be viable if we can really understand how to stabilize it in the cell and also have it still function appropriately.
Thank you. And then we have a second question from Martin. Thanks for the answer. You thought previous studies have shown that the bonds at the 3' end are not always stable, particularly in hepatocytes. I think this is something for Tom, I guess. Question to Tom. So I was just wondering. Because what you find is a quite a huge effect when you knock down XRN2. So is anything known about the function of XRN2? Because that might be also an important question to ask, right? Because if it has like a vital function, then it might be not worth knocking it down. So.
Right. Yeah, so it turns out that XRN2 is essential. So I didn't show any knockout data, right? And XRN2's sort of claim to fame is its role in polymerase 2 transcription termination. So at the end of termination, the RNA is cleaved, and then you have this sort of dangling 5' end of sort of runoff, and then XRN2 is supposed to chase down POL2 and sort of help inhibit that, right? We haven't tested, if that's being affected in our assays, POL2 transcription termination, but I agree. I think that's a very important consideration.
Yeah, and the other thing is, I think all or maybe most of your experiments were done in quite a relative easy setup, right? Just in the 293 T cells, so just embryonic kidney cells. So how is this mechanism if you look at other cell types or multiple cell types, or maybe more complex cell types?
Right. Good question. So I haven't looked at anything more complex than cell lines, but I have knocked down XRN2 in HeLa cells, A549 cells, I think HTT116 cells, and we still see an increase in these TRF1s. So it seems to be sort of a general effect.
Okay, that's good to know, because it might not be worth to continue on this. So any experiments in I think that's what you mentioned, right? So not more heterogeneous cell populations or something like this, or patient-derived cells.
Yeah, I think that would be a really great next step. But we have not done any sort of patient-derived cells or mice or anything like that.
Thank you. So I don't think there are any other questions in the chat yet. So I would like to thank both Brie and Tom for their excellent presentations. I really enjoyed both of them. And I would like to thank Hassan for co-chairing this webinar today. Thank you, Hassan. So if you want to give a presentation at these OTS webinar series, please contact me or Hassan or Erin, just using the OTS email address that you can find on the OTS webpage. Thank you for your attention, and see you in a few weeks.